All right, so I think we'll go ahead and get started. So first announcement, Carol Higginbotham, who's from chemistry, is going to be hanging out with us for today and on Tuesday to see how we do things in here. Some other announcements, so we have an in-class quiz coming up next Tuesday, so be sure to review material that we covered this week. And I'm going to show you guys, so for those that were able to participate in the tasting activity on Tuesday, and also for those that didn't participate, there's an extra credit opportunity. I'll show you guys where to find that. So you don't have to participate in the tasting activity in order to earn the extra credit, all right? For those that weren't able to make it to Tuesday and want to participate and experience Miraculent, I brought the stuff again, all right? And so what we'll do today is we'll go through and talk about eye, vision, and some of those related structures and the physiology, and then we'll probably get into a little bit of the ear in hearing, and then we'll, for the second half of the session, really kind of, the last part, we'll go and have that tasting activity. So if you guys want to stick around, you were able to witness it on Tuesday, you can stick around if you want. If not, feel free to take off. But remember, lab doors, if you have lab today, are going to be opening at noon. All right, so make sure you're back by then. All right, so like I said, we'll probably get into a little bit of the ear hearing today, but we'll finish that up on Tuesday. All right, so let me show you guys where to find some of the stuff on Blackboard that I just talked about. All right. So if you click on the ICA and HW, so if you guys are so inclined, you can find this extra credit here. It's called Miracle in My Mouth Extra Credit. All right? The directions you can find here, so be sure to follow them. Like I said, you don't have to have participated in the tasting activity to earn the extra credit, though. Okay? Also, if you go under the PowerPoints, there's a whole bunch of great stuff in here. Lots of extra resources, especially in the special senses. Some of these we'll take a look at them. Others, we don't have time to, but I think putting things in motion really helps with understanding the physiology of what's going on. So when you guys have run out of things to binge watch, right? So you guys can binge watch, some of these animations. Okay? All right, so there's a whole bunch here. And so we'll take a look at a few of them, in class, but we don't have time to take a look at all of them. The other thing I'll mention is that in the lab, for this week, there's working models of the ear and the eye. So this is one of the few opportunities where, paying attention to some of those working models helps you understand the physiology of what's going on here in lecture. And so make sure that, you take time to understand what's going on with those working models in the lab. All right. Any questions on any of that? Makes sense? Okay. All right. How about any questions on stuff we covered last Tuesday? For those that weren't here, that lecture I was able to capture and post up on Blackboard, so be sure to take a look at that when you can. Everything was clear, nothing fuzzy, hazy? No? All right. There's a few things that I don't think I mentioned, but I just wanted to circle back and make sure that I touch on, especially with olfaction. So we didn't talk about aging and smell, right? So what happens as we age with respect to smell? What do you guys think? Yeah, it gets duller, decreases. So remember these olfactory neurons? They get replaced over time, but it turns out as we age, they don't get replaced as frequently. And so overall what ends up happening is that you lose some of the ability to, smell certain things. The other thing is, how many folks have heard of nose blindness? So where you smell something and then someone comes in and says, wow, that really smells, but you don't smell it anymore? So you kind of gone nose blind to something. So it's kind of olfactory fatigue. So if we have, say, a corner of our room here, where there's a strong odorant, okay, and you're in the center of the room, at first you're able to smell it, right? Okay, then after a while, you no longer smell it anymore. But say you want to try to figure out from which direction that smell is coming from. So you walk around randomly, right? So eventually one of those random walks, you're going to encounter a stronger smell, right? and then you stay there for a second, and then you become nose blind again, right? Okay? And then you do that random walk again. Eventually, you start getting closer and closer to the origin of the scent. So think about in the wild. Think about in nature. So how would that be beneficial? To be able to track to a scent. So, what's that? Yeah, hunting, right? So being able to track, say, prey, right? So here we call that adaptation of the receptors. So here olfactory receptors, they adapt really quickly. One of the benefits of that is that you're able to figure out which direction that scent's coming from. All right? Does that make sense to folks? Everybody on board with that? Any questions on that? All right. Okay, so those were the two things I really wanted to make sure to mention about olfaction. And what we'll do is resume talking about vision and the eye. All right. So let's just start off with a quick review. Remember some of the eye structures we have. So we have our cornea, and then we have an area where we have some fluid. You guys will be able to see these things in the lab as well. And then we have the iris. The iris is the colored part of our eye. And so what gives us the color of the iris is melanin pigment. So individuals with lighter irises, don't have as much melanin pigment in them. All right? And then remember, the pupil is just that hole in the middle of the iris. And then as we go posterior, you get the lens, and then you have another really big chamber that's filled with a fluid. So anytime you see the word humor, you should think fluid, right? So we have vitreous humor. And so in the lab, y'all are going to be dissecting a steer eye, and you're going to be able to see how gelatinous this stuff is. All right? And then remember, there's 3 layers here. The outer layer was sclera. So it was tough. For those that had lab on Tuesday, when you're dissecting the eye, you found out how tough that layer is. Was it tough? Why is it tough? What do you say? So what do you guys think? So where do the extraocular muscles attach? To the sclera, right? So if you want to be able to move your eyeball around, you want to have it firmly attached to something. So that's one of the reasons the sclera is really tough. So as we go interior, what was that next layer? That was the choroid. So what did we find in the choroid? So the choroid. So underneath that, we have blood vessels, right? The choroid has the blood vessels. And then interior, the most internal one, was the retina, right? And the retina had the neural components. Right, had the photoreceptors, which is what we're going to talk about. All right, so here, one of the things that you're going to want to make sure to be able to do is describe, or at least name, the structures in order that light goes through. Okay, going from outside to inside of the eye. Okay, so we talked a little bit on the back of the retina. So we have the optic disc and the fovea. So we said the optic disc was a blind spot, right? And the reason it's a blind spot was because there weren't any photoreceptors. All it is axons of some of these cells. So do you guys see blind spots when you look around? Those are holes in your vision? No. So it turns out your brain, what it does is it kind of stitches things together, and it does a sophisticated, algorithm to try to, imagine, what would be there. Okay, so you don't see blind spots. And then the fovea, which is inside the macula lutea. So remember we said the macula lutea was like the target. The target, and the fovea was the bullseye in the target, right? This is where we have the highest resolution of images, right, of vision. Okay. And so here, if I go back, That's this area here. So is that jogging your guys' memory? Okay, good. Alrighty, so this is where we left off, physiology of vision. So we're looking at physiology of vision. How does it work? Turns out it's a bit backwards from the types of neurotransmission that we've talked about. So here, these cells are photoreceptor cells. And these cells aren't neurons, but they have specialized pigments in them. Specialized pigments absorb light. Okay? And one of these types of pigments, all right, are going to cause sodium and calcium channels to open. All right? So they stay open at rest in darkness. And then there is a inhibitory neurotransmitter inhibiting this second cell called a bipolar cell. So here in the darkness, right, this is constantly signaling this cell, okay, to put the brakes on, which means that the last cell here, which is actually finally our neuron here, isn't sending a transmission, right? Okay, it's got the brakes put on it. So here, no neurotransmitter is being released by the bipolar cell down here. So this ganglion cell isn't sending a signal along the optic nerve. All right? So how is this kind of backwards? Well, it's because in darkness, this constantly is sending a signal here. So normally in neurotransmission, you had to have the receptor being activated, and then you got a signal sent. Here we're saying that in darkness, right, we don't have very much light, and these receptors aren't sending, or they are sending a signal. Okay? So it turns out when light shines upon it, you basically taking the brake off the system. Okay? So it's kind of backwards from a little bit of what we talked about with neurotransmission. Everybody with me on that? So here in darkness, we're kind of putting the brakes on the system. And then in the light, what we do is we take the brakes off of the system. And so now we take the brakes off the system, which means that this bipolar cell releases its neurotransmitter, which causes an action potential in the ganglion cell, whose axon makes up the optic nerve and sends that information to the brain. All right? So here, this isn't darkness. There's 3 different cell types. And so here in light, what ends up happening is that we have one of those pigments that is going to be absorbing light, okay, retinol. So how many folks have heard that vitamin A is good for your eyes? So this is one of the reasons vitamin A is really good for your eyes, is because this retinol is a derivative of vitamin A. And it changes shape so that now this is no longer sending that braking signal to this bipolar cell. So the bipolar cell releases its neurotransmitter, which causes this ganglion cell to send the signal along their axon, which is making up the optic nerve. Everybody following me on that? Okay? All right. So I just want to kind of point out how these structures are laid out in the eye. So if we take a look at our eyeball, this is out of the way. All right, so here's our eyeball. Okay, so cornea, lens, and then we have our sclera, and then our choroid, and then the retina. Okay, so what I've done over here is kind of zoomed in on the retina in order. So here, the cell that is going to be towards the interior of the eyeball is the ganglion cell, okay, whose axon is going to go all the way down and then make up the optic nerve. So if I was to draw this out here, you got the axon of all the ganglion cells coming out here to make up the optic nerves. All right, Everybody following me so far on this? That bipolar cell that we saw is next furthest away from the interior of the eyeball. The one that's furthest away from the interior of the eyeball is the photoreceptor cell, rods or cones, which means that if we're tracking the pathway of light through the eyeball, right, first it goes through what structure? I thought I heard it. Cornea, right? And then we have this anterior chamber, and then what was this representing? Anybody guess? The lens, absolutely. So here are light coming along here. And goes through the cornea and then the lens. And here for the retina, it has to get past first the ganglion cell, then the bipolar cell, and then the photoreceptor cell finally will then get that light signal, right? So this is a bit counterintuitive. A lot of times students want to put the photoreceptor cell closest to where the light path is, right? But that's not how it's set up. It first is going to get past the ganglion cell, the bipolar cell, and then eventually reach that photoreceptor cell. Everybody with me on that? Okay, so it's a bit counterintuitive that light would have to pass through these cells with respect to the retina. Did you get a picture for Damien? Oh. Hold on, let me get this out of the way. Is everybody with me on that? No one fell off the eye physiology train? No? Okay, good. All right, so one of the other things, photoreceptor cell, the bipolar cell, they're not neuronal cells. They're not neurons. The only neuron here is the ganglion cell, which means it's only the ganglion cell that can generate an action potential, right? That action potential is going down along the axon, which makes up the optic nerve. All right? Okay. All right. So this is also another cartoon version of it. So you can see that we have our photoreceptor cells here. Here's our bipolar cells. Here's our ganglion cells whose axons are coming out, making up that optic nerve. Now you can see why the optic nerve, this optic disc area, we say it is a blind spot. Because are there any photoreceptor cells? No. What is it made up mostly of? What is it made up mostly of? So what part of the ganglion cell? So it's coming out here, sending the signal. Axon, absolutely. Okay? All right, so I'm going to play for you guys a little podcast. We'll cut out a little bit in the interest of time for the stuff that's, for some of the stuff in interest of time. If you want to hear the whole thing, you can.
So one of the sort of debates that became interesting to us is this: where is the color? Is it out there? Is this gray kind of right now? Is it red for everything? A B or L or anything? Does it exist in a way that you can pin down and say it's outside of you? Or does it only get to be red when it gets in the head?
Well, you can. Another more severe way to ask this, I ask this whenever I'm giving talks, is would aliens see it as red?
Yeah, would aliens see it as red?
Right. And the answer is almost surely no.
Here this says, Mark, even your dog would see it's red.
Your dog has color vision. It has blue, yellow, and black, white.
Really?
Yes.
So what would the world look like to a dog?
I mean, if you've ever known a guy who's colorblind, and 10% of men are colorblind, that's roughly what it's like.
Huh.
Well, here's a question. If a dog and a human and a crop were to be staring at a rainbow, would they be seeing very different things?
Yes.
This question that Robert was tossed out during the interview about how different creatures would see the rainbow, this ended up taking us down a little wormhole. We ended up actually getting the choir to help us illustrate what we learned. But just to set a baseline, your normal rainbow goes like this, starting from the bottom up. Red, orange, yellow, green, blue, violet, Roy-G bit.
I don't know why people put the INF, but that's
If you didn't have the info, you couldn't say it. You wouldn't.
that's why you need the INF.
That, by the way, is Tom Cronin.
I'm what's called a visual ecologist.
Mark suggested we give him a call. He told us that humans see 7 colors in the rainbow.
In the case of the dog,
Very different rainbow.
it's going to start off blue. He'll be able to see blue just fine.
So we would see a rainbow starting with blue,
Same blue we see.
and then grading off into
Green. Same green as us.
and then disappearing. Rainbow would happen there.
That's it.
Yeah, so the rainbow will only be that half as thick as ours.
Wow. That's a sucky rainbow.
That's why when God promised that he would never deliver another deluge, he made the promise in the rainbow. The dogs just were totally unimpressed.
And what is it about the dog eye that makes it see this way?
It doesn't have red sensitive photoreceptors, no red sensitive cone.
The weird thing is that the difference between dogs and us, cone-wise, is just one. They have cones tuned to blue and green, so do we. We have this one extra, red, which doesn't really seem like a big difference. I mean, it's just one cone. But
To have 3 is so much better than 2
that's Jay Knight's vision scientist.
because of this kind of multiplicative thing. Red can get mixed with blue,
Which makes purple.
or red can get mixed with yellow,
Which makes orange.
green can mix with blue.
Teal or turquoise.
That's how you get about 100 different shades of color in we can see. So by adding one photopigment, instead of adding just one more color, you actually add about 98 colors or so.
All right, let's move on. So now we have a crow.
All right, so you can listen to the middle part of this. I'm going to kind of fast forward to talking about one of the organisms that has a whole bunch of color cones. So the mantis shrimp. It's out there.
Butterfly has 5, depends on the butterfly. Mantis shrimp has 16. 16 kinds of receptors.
What would the rainbow look like to them?
Mantis shrimp would see the rainbow fine because they live in very shallow water, and so the water is pretty clear most likely there. They would start the rainbow way, way, way inside where we see violet. They would see extraordinarily deep ultraviolet. And then they would go on through several kinds of ultraviolet, probably 5 or 6 kinds of ultraviolet. And then we get to violet, probably reaching our colors, and go through violet and violet, blue and blue and blue green.
Where they have those blue, blues as well.
Yep. And then they would go out to the reds. They would be about as red as us when they got to the red.
But only in the red.
Yeah.
What a rainbow that must be.
Yeah.
They have the most complicated visual system of any animals, but factor 2.
Wait. You said any? You mean that unequivocally any?
Yeah, no other animal that we know of has a visual system within 50% as complicated.
All right, mantis.
But, you know, on the other hand, their brains are tiny, so who knows what we need to.
They may not have the ability to perceive the beauty of it.
No, I don't. No, they're mantis shrimps are into violence.
All right, I think that's about it. So we got 3 color cones. Mantis shrimp has 16, which is phenomenal. And the other interesting things about mantis shrimp, so Samson was telling me a few. So they're
snap their claws fast enough to push all the water out, flash the second.
mm-hmm.
So fast.
Yeah, it's a pretty amazing organism. All right, so did it make sense there? How just adding an extra red photoreceptor cone there, you can add, I think they said around 96. You can boost us up to about 96 different, colors and so forth. And so it's kind of just not simply additive, but multiplicative, the complicated nature of what we can perceive with colors. So here we've got 3, color cones, and we also have rod photoreceptors, which we use for night vision. All right. So here the cones require a lot more light, so they don't work as well at night. But the rod photoreceptors don't require as much, as much light. Okay. Let's see. Okay. So what about controlling light? We've talked about this a little bit already. So when we talked about the autonomic nervous system, we talked about this idea of controlling the pupil diameter. One, branch of the autonomic nervous system was a parasympathetic. The other one was a sympathetic, right? So here, parasympathetics involved with letting less light in. So if we're letting less light in, what's happening to the pupil diameter? Decreasing, right? So there's a muscle that is being activated that's causing that iris to close and therefore decreasing the pupil diameter. Because remember, the pupil is just a opening in the iris. Sympathetic, right? So sympathetic, when is the sympathetic nervous system activated? What kind of situations? Rest and digest we have. Is that true, sympathetic? What's that? High stress, yeah. Yeah, so remember, sympathetics, high stress, fight or flight, right? So here, sympathetics causing those muscles, different set of muscles to contract, causing that opening in the iris to expand, letting more light in, right? That's going to be helpful in case you're out hiking in the woods and you see a bear and you want to try to find an exit route, right? More light, better chance of finding an exit route, right? So here, the pupil diameter is controlled by the parasympathetic and sympathetic nervous system, which are divisions of the autonomic nervous system, right? And so here with vision, remember, it's really the optic nerve that's sending information to different parts of our brain for processing and then interpreting and identifying that information. Now, what word do we associate with that area of the brain if we're doing identification and interpretation of sensory information? Is that a primary or an association area? So association, absolutely, right? So primary is going to be mostly processing of that sensory information, right? Controlling the actual eye movement, right, are these 3 cranial nerves. So we've talked about these guys already. Extraocular muscles that attach to the sclera are controlled by these 3 cranial nerves. And you can see these on the models we have in the lab. So be sure to take a look at them. All right. Now, when we take a look at something far away versus close up, we have to make sure that what happens is that those light rays that get refracted, make sure that they converge on the retina. In order to do that, the lens can change shape. There's other things that also go on. All right? But here, when we are looking at something that is, say, far away, those muscles are relaxed and the lens is flattened. When you start looking at something that's close up, those muscles are going to contract and the lens is going to thicken. What this is doing is helping make sure that whatever you're looking at, those refracted light rays are going to be centered on the retina. Right? And this is called accommodation of the lens, or lens accommodation. Okay? And so it does that to accommodate for distances. Now, so here, the lens, as we age, layers get added onto the lens. It's kind of like layers of an onion. You keep adding layers as we age. And as that lens gets thicker and thicker, as we age, it becomes much more difficult to do this. This is a major reason why our eyes get worse as we age. We have more difficulty with doing this accommodation because the lens gets thicker and thicker. Yeah, go ahead. What do you mean? Oh, exactly, yeah. So if you're looking at something far off, these muscles that are called ciliary muscles are relaxed. And then when you're looking at, they're relaxed. And then when you're looking at something close, so if you're looking at something far away and then you look down at your piece of paper, right? What's happening? The lens is going to change its shape. And the reason it's doing that is so that it can make refracted light waves so that they focus on the retina. And we're going to talk about in just a second what happens if it doesn't focus on the retina. All right? So close vision, the lens bulges, and distant vision, the lens flattens. Does that make sense to folks? Everybody on board? People pretty comfortable? On a scale of 1 to 5, 5 being you can basically tell your parents and educate them about this. 1 being, I have no idea where I am. So, okay, good. I'm not seeing any 1s. Okay, good. Fantastic. All right, so not surprisingly, I wear glasses. A lot of people wear glasses. What ends up happening here? Well, turns out that with the refraction, sometimes that focal plane here, right? So this is where we don't have correction. That focal plane isn't at the retina. It's either past the retina or it is stopping short of the retina. Okay? And so how can we correct this? Well, you put a lens in front of our cornea. So our cornea actually does a lot of the refracting, right? So that you kind of pre-refract, these light waves, so that now the focal plane is at the retina. So for, people that are nearsighted, so like me, okay? So here, nearsighted, I can see things generally up close, okay. Things far away, not so much. And so my glasses, the lenses kind of look a bit like this. For those that are farsighted, right? There's a kind of a pre-refraction that goes on to correct. We're making sure that the light focuses on the retina. Is that making sense to folks? All right. All right, so here are kind of steps for how we're seeing things, right? So light rays, make sure that you know the sequence of structures that light rays pass through, right? You're going to want to make sure you know this. And so it's going to go through the cornea, a lot of refraction happens there, then through the pupil, and then we're talking about bending light, that's refraction, okay? And so here, we're going to need some sort of accommodation in order to focus those light rays on the retina. That's going to happen. The image gets focused on the retina. And you'll see in the working model of the eye in the lab that the image can be inverted. Okay? And the light rays are absorbed by those photoreceptors. Are the photoreceptors the first cells of the retina that the light goes through? No. Right? It goes through a ganglion and the bipolar cells and then finally hits the opsin, which are the pigments and the photoreceptors. Right? Okay, rods are involved with black and white. They can be activated with a lower intensity of light. Cones are involved with color. All right, so if you're the mantis shrimp, you got 16 different cones. You're awesome. Okay, and then here we have several synapses. Eventually, the ganglion cell is the one that has an axon that is going to be part of the optic nerve. So remember that ganglion cell is the only one generating an action potential. Everybody with me on that? All right. So you're going to want to know these sequence of events and the anatomical structures that light passes through. Now, we already talked about different parts of the brain and which lobe of the cerebrum has the vision association area of the primary vision. Occipital lobe, all the way in the back. Okay? So one of those resources I put up for you guys is called Secrets of the Mind, and part of that deals with something called blind sight, where individuals, they can't identify what the object is, but if you move it left or right or up or down, they know that they're moving, that object's moving left and right and up and down. It's pretty fascinating. There's other really interesting things in that documentary, which I posted up on Blackboard. All right. So just like you guys said, we got our right and left eyeball, all right, that is sending our cranial nerve to, and it gets routed. All right, there's a synapse here, and then it gets eventually passed on all the way to the occipital lobe of the cerebrum, where it is going to be processed and sent off to the visual association area for being interpreted. All right, and so here you can see that we have some crossing over that goes over, or happens, where the right part of the eye, you get some processing on the left part of the brain. Okay, so there's some decussation that goes on. Does that make sense to folks? So does that pathway make sense? Any questions on that? You guys comfortable with it? You look a little puzzled. Yeah, so here, if we track these, so here you can see kind of blue, and then you have red, so the red crosses over, and then it's processed over here, right? And then the same thing over here. So here you can see that the blue here. The blue crosses over and then is processed over here. So there's some crossing over. Remember, we call that decussation that goes on. Right? Cool. Any other? Is it clear? Fuzzy? Well, after reviewing it, if you guys have questions, bring them to class on Tuesday. We usually try to entertain questions about stuff that, we went over previously. All right? So here, processing of sight goes from the optic nerve, and then there's some processing that goes on in the midbrain and the thalamus. Okay? And remember, the thalamus, that's a major relay center for sensory information. So if there's anything you should remember about the thalamus, right, is that it is a relay center. Right? And so any sensory information that is going to be finding its way to the thalamus is going to be relayed and then passed on to the appropriate areas of the cerebrum for interpretation and identification. Right? And like you guys said, this whole thing is happening in the occipital lobe, the visual association area and primary visual cortex. Right? Both of these are located in the occipital lobe here. Okay, so I just wanted to mention a few things. We don't have enough time to go into all of the interesting things that happen with the maladies of all these systems that we talk about, so I'll just mention a few common ones. One of the problems with the eye is cataracts. All right, so cataracts are clouding of the lens. Right? And so you can see this kind of milkiness, cloudiness of the lens that happens. How do you think that is manifesting itself in individuals? So what does that look like in individuals? If you had a patient that came in, right? What would they say if they had a cataract? What would they describe? Yeah. Blurry vision, that's the main one, right? Blurry vision. Now, are they going to have blurry vision when they look at something far away and up close? Absolutely. Okay, so the other one is glaucoma. How many folks have heard of glaucoma? Right? Does anyone know what that is? No? So it turns out that glaucoma is when the pressure within the eye is elevated. So if the pressure in the eye, so let me go back a few slides, quite a few slides actually. Right? So if we get a whole lot of pressure, right, built up in this chamber, it's going to push everything. And if it pushes everything, it pushes it kind of out of balance, right? And if we get a lot of pressure built up, it can be pushing pressure on some very sensitive tissues that are important for relaying sensory information with regard to vision. And so individuals with glaucoma, one of the major ways is to try to relieve that pressure by increasing or decreasing blood flow to that area, increasing drainage, right? Okay. So glaucoma has to do with elevated pressure, and that elevated pressure, intraocular pressure we say, is going to be pushing up against some sensitive structures. Okay. So I just want to go over some of the anatomy for the ear, and then we'll switch over to doing the tasting activity. All right? Okay? All right? So the ear turns out the anatomical structures and some of the physiology for how we hear and do equilibrium. It sounds simple, but the structures in the ear, I think, are really challenging because they're three-dimensional. And so spend some time in the lab with looking at the ear model. It's going to help you a lot with understanding how the ear functions. So we're going to explore here in lecture. All right. So what we're going to do is talk about the anatomy and then the mechanics of sound, so how we hear. And we're going to talk about balance, equilibrium. There's kind of two versions of that, static and dynamic. So this is also known as kind of linear, and this is angular, so if you're spinning around. So those are that's the approach that we're going to take. And so I just want to talk a little bit about ear anatomy, and we'll finish up with talking about the physiology next Tuesday. All right, so we can divide up the ear into three regions. One is the external ear, the middle ear, and the inner ear. All right, so what divides the external ear from the middle ear is the tympanic membrane. Here we have the ear canal. So what's the ear canal have in it? What's that? Earwax. Hopefully it's not filled with earwax, right? Okay, so here you have maybe some earwax, but hopefully it's mostly air. All right? What about on the other side of the tympanic membrane here? This kind of opening, this tympanic cavity. It's also filled with air. Okay? So it's filled with air. Now there's a tube that leads down here. So when I learned about this tube, it was called the eustachian tube. Then it was called the auditory tube. But people got confused between that tube and this tube. And so they came up with another name, pharyngotympanic tube. All right? Now, I'm pretty sure the ulterior motive was to make the A&P students have to study more stuff, right? So here, why is it called the pharyngotympanic tube? Well, it goes to the tympanic cavity and then all the way to the pharynx. The pharynx is the back of the nose, the nasopharynx. So it empties out to the back of the nose. And it is a pressure regulator. It regulates the pressure that's found on this side of the tympanic membrane. So when you guys are driving up and down mountains, you can hear that popping sound, right? That popping sound is this tube quickly opening and closing to relieve pressure, to make sure that the pressure on this side of the tympanic membrane is the same on this side. Okay. No, this tube. This tube. Yeah, this tube. Now, the ossicles, tiny, tiny bones, which we have in the lab, right? Those are the malleus, the incus, and the stapes. So here the tympanic membrane looks like kind of the surface of a drum. And when air waves hit it, it causes this bone to cause the next bone, the incus, to move, that causes the stapes to move, right? Now, up to this point, we said that these kind of openings, these cavities, were all filled with air. Here, the inner ear is filled with a liquid. It's called perilymph or endolymph. There's two types. Okay? So here, this is liquid filled. This has air. Okay? So it has gas. Right? Now, here the stapes is over something called the oval window. And then we also have something called the round window here. Okay? So this whole fluid-filled chamber starts at the oval window, and we'll see what the cochlea ends at the round window. You can see this in the model in the lab. This cochlea, what kind of shell does that remind you of? Snail shell, right? So that's what it refers to. This is where we have hearing going on, is in the cochlea. The equilibrium that we talked about, the balance, happens up here with the semicircular canals here, and then also the vestibule. So here's our equilibrium part in the inner ear, and then we have the hearing part of the inner ear. Okay? So just some basic anatomical structures. So remember that cranial nerve that is sending sensory information from the ear? It was called the vestibulocochlear. Now you know why it is called the vestibulocochlear, right? Because it's getting information about equilibrium and it's getting information about hearing. Okay, so that cranial nerve, the reason it's named is because it's getting information from those two anatomical regions. All right? And so you take a look at that middle ear. We have the ossicles, like I said, the tympanic membrane. Then you have the malleus, the incus, and the stapes, which is covering the oval window. And so all of this in the inner ear is all fluid filled. All fluid filled. Okay? And here you can see they named this the auditory tube. That's the pharyngotympanic tube or eustachian tube. All right? Okay. So this will be the last slide, and we'll take a break and then do the tasting activity. All right? So here, we're going to first take a look at the inner ear. We have the cochlea, which is involved with hearing, and then we have the vestibules, okay, and then the semicircular canals. This is involved with equilibrium or balance. Now, for looking at this cochlea, all right, it's a complicated structure, and so I'm going to show you a little bit about looking at this video on hearing to kind of help explain what it looks like.
Hearing occurs when sound waves are changed.
Of the inner ear. When the eardrum vibrates from sound waves, a chain of three tiny middle ear bones moves. The malleus, incus, and stapes, or hammer, anvil, and stirrup, together called auditory ossicles. The arrangement of these bones forms a lever system that amplifies the energy of the sound waves before it reaches the cochlea. The stapes fits into the membrane-covered oval window at the base of the cochlea. Within the bony cochlea is the cochlear duct. A Alongside the cochlear duct are the vestibular duct and the tympanic duct. All three ducts are filled with fluid, endolymph in the cochlear duct, and perilymph in both the vestibular duct and tympanic duct. When the scabies pushes against the oval window, pressure waves in the perilymph travel up the spiral through the vestibular duct and back down in the tympanic duct. Pressure is relieved when the wave reaches the round window. When looking at a cross-section of the cochlear duct, specialized hair cells are shown being struck by pressure waves in the perilymph. The endolymph-filled cochlear duct now appears as the central triangular section. The vestibular duct and tympanic duct, both containing perilymph, are on either side of it. Sitting on the basilar membrane of the cochlear duct are hair cells of the organ of Purkinje. Above the hair cells is the tectoral membrane. When the basilar membrane is moved by pressure waves in the perilymph, hair cells are pressed against the tectoral membrane, and their stereocilia are distorted. The distortion generates a signal that travels through the cochlear branch of the vestibular cochlear nerve to the brain, where the signal is ultimately interpreted